Welcome to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Eva Amsen, and experts in the field of cryo-electron microscopy.
Today on Cryo-Talk, we're joined by Liz Kellogg, Assistant Professor in Molecular Biology and Genetics at Cornell University. We're talking about her research.
We are really interested not just in understanding the mechanism of these CRISPR-associated transposons, but in adapting them as genome engineering tools.
Her advice for early career researchers.
You are the best judge of what is best for you. So have confidence in yourself.
And what kind of music she listens to.
I listen to a lot of 2000s pop because that was my time,
All in this episode of Cryo-Talk. Hi, and welcome to Cryo-Talk. I'm Eva Amsen, and I'm here today with Liz Kellogg, Assistant Professor in the Department of Molecular Biology and Genetics at Cornell University. Her research group uses cryoEM to study molecular mechanisms related to DNA recombination and protein engineering. So Liz, how are you today?
I'm good, thank you.
Thanks for joining us. Now, we always ask our guests to share a little bit of their research backstory, and I think you have a connection to one of our previous podcast guests. Yeah. Can you tell me a bit?
Yeah, right. So I started my PhD. I did my PhD with David Baker at the University of Washington, and there I worked on protein structure prediction techniques. So I did a purely theoretical PhD. And then I moved to Berkeley, where I did a postdoc with Eva Nogales, who was your previous guest and one of my favorite people. There I studied microtubule structure using cryoEM, and that was around 2012 or 2013. That's about the time when the direct detectors came online. So I was very fortunate in the sense that I never had to collect data on CCDs or film. I went straight to the direct detectors, and of course, we were astounded by everything that we could see at that time. We made a number of different discoveries regarding microtubule structure, as well as the proteins that bind microtubules and affect their function, things like tau. So tau is a protein important in Alzheimer's, that is a factor in Alzheimer's disease. And then actually, to cap off my postdoctoral studies, I started moving into studying recombinases, and that has really set me on the path for my research group here at Cornell.
Yeah, because you're looking at mechanisms that could be useful for genome engineering. Can you tell me a bit about that?
Yeah, so we're actually very interested in things called transposons. Transposons are mobile genetic elements. They're selfish genes that essentially replicate their own DNA. And so we were very interested in their ability to cut and paste DNA without introducing these, while bypassing double-strand breaks. They essentially affect this process. They do this autonomously. But the class of transposon we're extremely interested in now is called CRISPR-associated transposons. And that means that these transposons naturally associate with a CRISPR effector. These effectors do not cleave DNA. They actually bind the RNA and the target DNA, and they recruit the transposition machinery to that site for insertion of their genomic payload. And so a lot of people are very interested in using these systems as tools for programmable DNA insertion. It's very exciting, and we're continually astounded by what we learn. These transposons end up being very complicated mechanistically speaking, and so a big push in my lab is to uncover the molecular mechanisms utilized by these CRISPR-associated transposons using cryoEM.
Yeah, so that actually leads me right into my next question. That's how are you using cryoEM for that?
Yeah, so cryoEM is great as a tool, as you well know, speaking to our previous guests, because you don't need to crystallize your target of interest. So we can actually reconstitute large ensembles of proteins onto DNA substrates. Now that the challenge is to figure out what DNA substrate to use in order to stabilize the full assembly. But we have found that cryo-EM is incredibly useful in terms of unraveling these mechanisms because we can visualize different states. One of our previous papers that was published in Science in 2021 really was digging into the structure function relationship of a AAA plus regulator that was involved in the programmable DNA insertion cycle of this transposon. And so we characterize this protein in a number of different states and with its other transposition related factors in order to try to understand how this process was occurring. And that's not possible using any other technique, really.
Yeah, it's really it's one of those techniques that it makes you be able to look at things that you can't do in any other way, which is kind of what we've heard in every episode so far.
Yeah, we love
it. So if you could look into the future of cryo-EM, what do you think, where do you think it will be in like 5 to 10 years, maybe thinking about the kind of applications that people are using it for?
Yeah, I think that's a really good question. I loved the previous episode with Eva Nogales. I thought her answer was really great. Her answer was complexity. And I would say, yes, that's absolutely the right answer. But also, one thing that really excites me about cryo-EM is its ability, like I said, to characterize protein structures that are not in the crystal state. So they're essentially doing whatever it is they do in solution, you're flash freezing that, and then you're able to see that in an electron microscope. And I think, that was something that we had dreamed of modeling back when I was in the Baker lab. That's very challenging. And I think like the future of cryo-EM is in visualizing not just one stable ground structure, but, the transient states and the dynamics that are contained within that thermodynamic ensemble. And so that's what really excites me about cryo-EM and for the future. I think that there's a lot of work that needs to be done before we get there, but that's what makes this so exciting.
Yeah, definitely.
Yeah.
And you started your lab in 2019, so it's not that long ago. How have those first few years been for you? And what are you looking forward to doing in the next few years?
Yeah, that's a really great question as well. So starting in 2019, I of course was affected by the pandemic, like every other assistant professor. That's been exceedingly challenging. And I think, in 2020, when things shut down, it was hard because, we had to keep our growing labs together. You had to maintain some sort of cohesiveness when everybody was alone at home by themselves. I think that was very challenging. But right now is still challenging as well for growing labs because of supply chain issues. There is turmoil in the job markets. And so I think that's something that's kind of that I reflect on a lot with me and my peers is how do we help one another launch our labs while maintaining a great lab culture and doing cutting edge science. We talk about that a lot. And furthermore, I would urge anyone who's listening in the more senior positions, it's like the pandemic is going to echo throughout the years. It's not just this 1 year that, we all got tenure clock extensions for. This is going to continue on impacting all of the young assistant professors. And so I would really urge more senior colleagues to keep that in mind when people come up for evaluation.
Yeah, that's definitely something to think about. And I guess also, working from home is not possible if you work in a lab. So it's you'll have to you're competing with them, like PhD students deciding whether they want to be in a lab or do something else. They see their friends going into places where they get to work from home, and they might have to not do that.
Also, I don't have small children, but a lot of my colleagues with this is also the time when people start families. And I think that was like, I don't have children, but I can't even imagine, that must have been so difficult. So, that's even without the additional challenges of having small children. So,
what are your plans for the next few years, assuming no further pandemics?
gosh, I would love it if we could have like, returned back to the way things were, but I don't know that that's going to happen anytime soon. So we are really interested not just in understanding the mechanism of these CRISPR-associated transposons, but in adapting them as genome engineering tools. So I would really like to, in the next coming years, to combine my training that I got from the Baker Lab in protein design, protein modeling and design, as well as my training in cryo-EM. So I would really like to meld those two. We are really very quickly gaining very good mechanistic understanding of how these elements work. And once we, establish that foundation, I think then the question becomes, how do we adapt these tools for practical purposes? So we're very interested in going in that direction.
That sounds
There's
exciting.
So many things I would love to do, actually.
Yeah.
It's just not enough time to like talk about all of them, but there's so many things, and that's one of them. But yeah, I'll just stop there.
And what about when you're not working? Do you have any hobbies?
So I used to do ballet very seriously, but it's been challenging to fit classes in while I'm trying to launch a lab. I also started horseback riding. That's one of the nice things about Ithaca, is that, you get access to all sorts of kind of country activities that you may not have access to otherwise. And I also run now. So in order to kind of keep in shape, because mental health is just as important as physical health. So I try to combine hobbies with exercise.
Yeah, that's a good plan.
Yeah.
Yeah, and you mentioned the countryside already, but that's I think we're going into our round of quick fire questions, and that's actually my first question on the list is, do you prefer the city or the countryside?
I think they both have their merits. I like both. I think that I'm the kind of person that when I'm in the countryside, I like to be in the city, and when I'm in the city, I like to be in the country. So it's hard to say. I would say both are good and bad in their own ways.
And do you like to cook?
I love cooking. I love it. And I love to improvise. I love to just kind of go into my pantry and take out whatever I have and then make a delicious meal.
Any favorite recipes you want to share?
My go-to is salsa verde chilaquiles. I usually make that on Saturday mornings.
And do you like reading? Do you have any book recommendations?
I loved reading. I don't read as much as I read a lot of papers. I don't read a lot of books, although I really would love to get back into it. The last book I read was Bad Blood. And this was the book by John Carreyrou about the Theranos story. That's the last novel I read. So.
Sounds interesting. I also kind of want to see the show about that.
Fascinating. Do it.
Speaking of shows, are there any films or TV shows that you watched in the last year or two that you would recommend?
It's a really good question, and actually nothing really pops into my mind at the moment. There's a movie that I've been wanting to see, which is Everything Everywhere All at Once.
Oh yeah, I've been meaning to see that. I also still need to see it.
Yeah.
Maybe by the time this airs, we'll have both seen it, but
I hope so. It looks like a really good one.
Yeah. And do you listen to music?
All the time. Yeah.
Like, what do you listen to?
I love pop music, so I listen to a lot of like 2000s pop because that was my time, a lot of Katy Perry, and let's see, Nicki Minaj, stuff like that.
So do you listen to music while working, or just while you're running, or all the time?
All the time. I listen to music and I listen to podcasts. And I've listened to the Cryo-Talk podcast as well.
Of course. Yeah. And if you were not a scientist, what would you be?
Yeah, that's an interesting question, because I actually, with my colleagues, we sometimes think about this, but I think I would be a writer, because I actually love writing. So I think I don't know if I would, seems like a big change, writing scientific manuscripts to novels, but I have a couple ideas.
Well, you can do both, as we've learned from our first podcast guest,
Yeah.
Joachim Frank. So he's
He's
managed to do
incredible.
both, so it's
Wow.
possible.
He's so incredible. Maybe later, after I win the Nobel Prize.
You got to do that first, and then
Yeah.
And something else that you mentioned earlier is you also have a podcast. Do you maybe want to tell people about that?
Sure. Yeah. Thanks for giving me the opportunity. So the podcast that I developed along with Mike Chinfroco and Mimi Ho is called The Plunge. And we actually interviewed 6 different members of the cryoEM community. And we asked them things that, we had always wondered, things like, your most significant discovery, how did you feel about that? What is the future of cryoEM, as well as, more practical things that we wonder about for ourselves, like how do you create a thriving lab culture? How do you mentor the next generation of students effectively? Things like that. So I had a lot of fun doing it. I hope that people enjoy listening to it and watching it. So, but we'll see.
Yeah. And now you get to be a guest here. So
thank you so much. Yes.
Yeah. So I've got one more question for you. And do you have any advice for researchers who are just starting out their career?
Yeah. I also think about this a lot too. I mean, having advised students, I think a lot about how best to guide them. I kind of struggle with this because I feel like everybody's trajectory is so unique. And what works well for me, someone like me, may not work very well for someone else. So I guess personally, what I've found is that in my career, I would ask many people, all my mentors and more senior colleagues for advice. And you get all sorts of different advice about what's best to do. And I think what I realize is that everybody's giving advice based on their own experiences, about the challenges that they encountered at the time that they were trainees or they were at that particular stage. So what I always tell trainees is that it's good to seek advice, and you should. But keep in mind that, you are the best judge of what is best for you. So have confidence in yourself. And ultimately, you're the one who gets to decide, So, you can get advice, but I think that, have confidence in your own sense of direction about where to go next.
Yeah, that makes sense, because everyone is different. And
I mean, you're the one that knows best about what you like, what you enjoy, and where where you should go next, what you should do next, things like that, right? So
yeah. I always personally find it hard when people like ask me for advice, because what do you want me to tell you what I did? Because that's the only experience I have. But yeah,
exactly. I have a very, very limited,
Yeah, I've done one thing. That brings us to the end of today's episode. Thanks everyone for listening to or watching Cryo-Talk.
Thank you so much for having me.
Thank you for listening to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/cryotalk.